In the last chapter, we are going to talk about some molecular methods for diagnosis of genetic diseases. We use molecular diagnostic methods to detect inherited genetic diseases or some acquired conditions such as malignancies. Molecular methods can be performed on samples taken from fetus during pregnancy or can be obtained after birth. At prenatal stage, we can extract amniotic fluid or fetal DNA from maternal blood, and also we can take a biopsy from chorionic villi to obtain a sample for molecular analysis. These are identical to the baby cells and contain the same DNA and chromosome. In postnatal stage, peripheral blood lymphocytes are suitable for sampling. Some of the important indications to perform prenatal cytogenetic diagnosis, including advanced maternal age, confirm some carrier status, fetal abnormality detected through ultrasound examination, abnormal results on routine maternal blood screening, family history for chromosomal anomalies such as previous affected child, and determination of the fetal sex when the parent is confirmed carrier of an X-linked disorders. Here you can see the indications of postnatal genetic analysis include baby with multiple congenital anomalies, suspicious of having metabolic syndromes, the child with unexplained mental retardation or developmental delay, child with features of aneuploidy such as Down syndrome or Turner syndrome, and also other chromosomal abnormalities such as deletion or inversion. Molecular diagnostics are used to identify somatic mutation in cancers and also to identify infectious diseases such as tuberculosis, influenza virus, or specific strains such as H1N1 virus. Also, by these methods, we can monitor the treatment efficacy using assay for viral load, or for some cancers, target therapy is developed. In situ hybridization is a procedure that allows to locate the positions of specific DNA sequences on chromosomes. Today, most in situ hybridization procedures use fluorescent probes to detect DNA sequences, and this process is referred to as FISH. This technique is used to diagnose many types of chromosomal abnormalities such as numeric abnormalities, subtle microdeletion, complex translocation which cannot be identified by routine karyotyping, and gene amplification. All the samples which we talked about in previous slides can be used for this method. In molecular hybridization, a labeled DNA or RNA sequence is used as a probe to identify the target. Before hybridization, the DNA probe is labeled by fluorophore. The DNA probe then is incubated with a full set of chromosomes which have been attached to a glass microscope slide. The label probe and the target DNA are denaturated by heat or chemicals. The probe specifically hybridized to its complementary sequence on the chromosome and can be detected using fluorescent microscope. As I said, before hybridization, the target and the probe, which is labeled by fluorophore, must be denaturated by heating or chemical agents and then mixed together. So the probe hybridized to its complementary sequence on the chromosome, and that can be visualized by fluorescent microscope. In FISH technique, we need previous knowledge about the target chromosome region that is suspected of being altered, and we are going to detect it. Hybridization is the process of combining two complementary single-stranded DNA or RNA and allowing them to form a single double-stranded molecule through base pairing. Hybridization is a part of many important laboratory techniques. The genome is the entire set of genetic instruction found in a cell. In humans, the genome consists of 23 pairs of chromosomes. Comparative genomic hybridization is a technique that allows the detection of chromosomal copy number change without the need for cell culturing. It provides a global overview of chromosomal gains and losses throughout the whole genome of impaired cell. Routinely, sample DNA is labeled with green and normal reference DNA is labeled with red, and they mix with same ratio and hybridize to normal human metaphase chromosome spread, which are located on the slide. The green and red labeled DNA fragments compete for hybridization to their locus of origin on the chromosomes. The normal reference DNA and the metaphase chromosome are obtained from a healthy volunteer. The green to red fluorescence ratio measured along the chromosomal axis represent loss or gain of the genetic material in the impaired cell at that specific locus, and they can be analyzed by a computer. A higher intensity of the test sample color in specific region of chromosome indicate the gain of material of that region in the source sample, while a higher intensity of the reference sample color indicates the loss of material. A yellow or neutral color indicates no difference between the samples in that location. Let's review it on this slide. The test sample DNA is labeled with green and the reference DNA is labeled with red fluorophore. Then they mix with equal ratio and hybridize to normal human metaphase chromosomes. The yellow color means that the material of the specific region is balanced. When the excess material is present in the source sample, green color is detected, while the loss of material will produce red color, which is indicated of reference DNA color. PCR is a method used in molecular biology to amplify a very small sample of DNA to a large enough amount to study on it. The DNA is amplified in cycles of temperature change. In the PCR setup, several components should be included. DNA target region to amplify, for example, virus DNA. DNA polymerase that synthesize new DNA strand. Two primers, which are short single-strand DNA fragments that are complementary sequence to the target DNA region. Nucleotides to form a DNA strands called DNTP or deoxynucleoside triphosphate. Buffer solution, a chemical environment for optimal activity and stability of the DNA polymerase. Thermal cycling exposes reactants to repeated cycle of heating and cooling. DNA polymerase can only bind to a double-stranded region of DNA. Primers hybrid with a target DNA and mark an initiation site for DNA polymerase to bind. Each PCR cycle includes denaturation, annealing, and extension or elongation. In denaturation, the double-stranded DNA is converted to two single-stranded molecules by heating. Then, in annealing, the temperature is decreased that allowing the primers to attach to each of the single-stranded DNA. In extension, the DNA polymerase finds the primer and synthesizes a new DNA strand using free nucleotides which are present in buffer solution. You can watch the PCR cycles in next slide.